So I wanted to just show you the locations of the Swedish University of Agricultural Sciences. So we are a little bit similar to INRE in a way, because we have three main campus at three different locations in Sweden. But we also have a smaller campus elsewhere as well. And the one where I'm working is here. This is.
Would you love this picture?
So how is it in Zoom? They will not see me.
No, they will not see.
I'll try not to point too much.
Yeah.
Strange.
We'll get used to it.
So we work mainly at our department with microbiology, molecular biology, and biochemistry and food science. That's our main topics. In the south, they work more with agricultural, maybe cultivation and so on. We have also up here some more fisheries and so on. So a little bit divisions, but we have veterinaries in Uppsala also. So divided, but we have a lot of cooperations in between different groups. So just want to say a few words about our group where I work. And there was this person actually gathering all the keywords that we have been using since 2016. And you can see by color how they have changed a little bit. But it's like our main topic is definitely anaerobic digestion and biogas production. And a lot of parameters connected to that. And syntrophic acetate oxidation has always been, since many years, been a main topic for us. And some new words are plug flow reactors. We have microbial communities. I mean, that has also been a keyword for a long time, but maybe we are changing the wording a little bit, but this is the main topics for us. And it's been really nice to walk around and hear your our talks and so on, and see the lab, because I think we have a lot in common here. So it's been really nice. And just wanted to show some of the reactors we have. Of course, we have CSTRs, as you also have, as well plug flow. We were talking about problems with plug flow and animal manure in the car. Now it's we know how it is when reactors explode. I was telling about these small windows that we thought was really like clever, that we could see the sludge, but when we had some stops in the feeding tube, then the windows just exploded out. It wasn't that clever. So these ones we have removed. Have windows in the reactor. We have just constructed a syngas reactor where we're gonna convert syngas to V-phase. And we do a lot of anaerobic cultivations. And we also have some like the main topics we have been working with for a long time. We have done a lot of microbial community analysis with cultivations and molecular analysis. And we do a lot of isolation and characterization of novel species. But this is always sort of a parallel work, because it's hard to get fundings for saying, well, we want to isolate new species. And they're like, yeah, why, more or less. So this is, I mean, something we really want to do, and I think is quite important to do still, to bring research further. So we try to do it like on spare time. We have a new project working with biogas degradation and biogas reduction together with an EU project with Dominic and okay. And we also have some persons working with antibiotic resistant genes in AD, and I heard that some of you are also doing that, so that's nice. But I was planning to focus on the syntrophic acetate oxidation today. in order not to have too long talk. So my interest I did my PhD working with syntrophic acetate oxidation at high-ammonia. But my eye-opener for propionate was actually in 2015. And I checked your publications. I think we had some propionate issues and during the same time, when this is a reactor where we changed temperature and then we increased the loading. And then we saw, well, of course, we get acetate, but with the higher ammonia, we also get propionate problems. And I wanted to see who's in there and who's degrading the propionate, who can we support them. But then I found out that I couldn't find any, known species doing this. So I thought that we need to look into this more. And this, all of you already know that we have inhibition of the acetoclastic methanogens. So we take another route to degrade acetate and to hydrogen carbon side to methane. But also when we have propionate, we will have acetate form. So this formed acetate also needs to be converted somehow. And so, well, yeah, then you need two syntrophs working together. And who is really degrading the hydrogen carbon side then? Is it the same methanogen? Is there different? And so on. So these kind of questions I had when I started to work with this. And yeah, so of course, you know that they are really dependent on each other, and there are a lot of different definitions of what is syntrophy. But I like this where we say that there are different chemicals formed when they work together compared to when they work separately. So you were a little bit discussing this yesterday. Is it syntrophy if their hydrogen is consumed? Well, yeah, that's a definition. It's not always really clear if they are really cooperating, if they are partners, and it's also often very difficult to know. But what we can say is that this syntrophy is often at cost of low energy gain, and they have slow growth rates. And they rely on hydrogen format transfer or direct electron transfer. But now we have this new paper out, there's a new paper out this last year, in December, where they show that actually methanol can be the fourth mode of syntrophy, which is really interesting. So things are happening in this area, and I think it's very, very nice that we see that there is openers in this field as well. Shouldn't be too strongly. We need to have our open minds because they are so diverse and need to find the ways to get rid of the electrons. So I think they try to use whatever is possible. But it is a challenge for us to sort of, to really show that this is happening. So this has been the classical mode of identify the syntrophs. So you have your reactors, your conditions, you do enrichments, and then you do isolations. And if you're really, really lucky, then you will get a colony, which is the right bacteria, and then you get the pure culture. And if you're really, really lucky, then you get the co-culture. And there's so many steps on this road that will hinder you. So there's a lot of luck here, let's say. And it's a lot of time. These are years of work to do this. So I thought, well, we should maybe try to go another direction, because also when you create this pure culture, you break the connection between the bacteria and the methanogen. So it can be really tough to connect it again. And also you might miss something by doing that. There might be some interactions that you lose by doing this. So I thought that, well, if we start with just identify who they are, we can work more with transcriptomics, try to find their characteristics, and try to optimize their activity to make them degrade the acids faster. And hopefully in the future, we can move back to your biogas reactor again. And as I was saying, this is in parallel. We try to isolate and try to get this pure culture, because if you want to really give them a name, you need that, of course. But in between, in order to get any papers out or any grants, yeah, this was instead the main workflow. So I got the grants and managed to start my first reactors in 2017, working in these smaller ones. So we inoculated with sludge from high-ammonia digesters and fed them with a medium containing propionate and acetate to have something to compare with compared to the acetate. In this medium, we have the yeast extract, because it can be quite difficult to cultivate them without that. And we kept the ammonium high during the feeding, so that we had ammonium in the medium. And we managed to identify the ones that we were only in the propionate reactors, and we saw that they belonged to the same family, but there were different genera. And you can see, even though we feed them with only propionate as a carbon source, in addition to very low levels of yeast extract, I mean, they're really low in relative abundance. And this is something you also see when you do, like in the biogas reactor. You seldom see that, yeah, because they are so low in number of species. But they have a key role, so you shouldn't forget about them, even so. So we have sequenced their genomes, and they are, this is the mesophilic, and this is the thermophilic. Quite similar, but still different. But they are among other syntrophic propionate oxidizing bacteria. So they're related, that it seems that they have some specific functions. We also managed to identify a new ammonia-tolerant methanogene that was a partner for them, which is a methanocoleus. But in order to try to enrich them further, we have removed the yeast extract now in the medium. And then we can see, well, so we only add propionate as a carbon source. We have a little bit of cysteine in our medium to keep the oxygen level low. So we think we have some species maybe growing on cysteine, but still now it's only the propionate present. And this is propionate fed to, this is a batch culture. And we can see, like, this is the syntrophic acetate oxidizing bacteria, the green one. And we have it's not super easy to see, but the beige is the syntrophic propionate oxidizer. But we also have this red one, which is quite surprising. It comes up. And it's an acetomicrobium. And we see the same thing in the acetate oxidizing community, that we actually have a species, we have this acetomicrobium, but we also have this alkalophilus coming up, actually when we spike with acetate. So this is quite surprising, because it doesn't have the pathway used for acetate oxidation that we know of. So when we have done metatranscriptomic, we see that we have the propionate oxidizer, sends out the acetate using syntrophic acetate oxidizers, and they both send out formate and hydrogen, or they use these compounds to send it out to the methanogene. But then we have this additional species doing something. And I know there are some persons looking into the biogas digesters that have seen them as well, and that they use the glycine cleavage pathway. So the idea is that it might use formate that's sent out from the syntrophic bacteria, or it might use acetate. But it's so difficult to know because we don't know in which direction the pathway is taking. But we also see some activity connected to amino acids, degradation or synthesis or something. So yeah, it's very, very tricky. And I mean, we have such a restricted community, and we can't still tell from our metatranscriptomic what it is doing. So it's very tricky. Please ask questions if you have now during the. It's super fine. You can do that now. So this is an open question still that we have, that we will look into further. And I can just show you. I mean, it's not like it's taking over the culture or anything, but it has activity, even though they only have the assets to grow on. So we are waiting for your modeling site to tell us the possibilities of these additional species. There is also been suggested by teams in I think they're in Japan, that they can synchronize their biosynthesis of amino acids, so they can sort of help each other to, if there are amino acids that are quite expensive to synthesize, they can sort of share them between them, because that can help them to survive during stress conditions. And since we have them growing without yeast extract, that's an option. We thought that, well, that could be something to look into and can be quite interesting to show. Maybe it's not only the syntrophy that they share the hydrogen and formate. They might even have like some cross-feeding of amino acids. But it's also a little bit tricky to sort of see that, because we thought, well, maybe, well, if we look at the acetate oxidation community and compare with the propionate oxidation community, what is the impact that you have this SPOB? And methanion, it didn't care at all. We saw some downregulation of genes connected to some amino acids from the SAUB. But then we realized, actually, just by having propionate in your medium, this can be the case. So this is also something, a question that we will keep on looking into. And we have tried to add some different amino acids, trying to see if this is something that can help them to grow faster. Please notice that these are days. So I have very patient PhD students. We have to plan our projects quite far ahead. We should not have like a vacation. I mean, you can shorten the degradation maybe 20 days by adding some amino acids. We have the control, the black one here. So, for instance, aspartic acid can be quite beneficial for them. So then this next question, this is just like growth, the cultures. So we are going to look into the sort of metatranscriptomic later. And again, I think the modeling can help a little bit here, because it can be quite complex. And then we have the part about the cell distance, because since they are cooperating a lot, rate limiting step is the transfer of the electrons between the bacteria to the methanion. And this is how they look, the mesophilic cultures. when they grow. So we thought, well, this is so interesting. They need to form flocks, and that must be like a key feature that they have in order to be able to degrade the acids and make them stronger. Maybe it is even a feature that helped them to tolerate the ammonia better. So we decided to look into this further. And I have my PhD student, he sort of constructed this thing. I don't know what you call it, but this is a camera here. So it moves under here and take pictures of these cultures over time. And I have a movie. I hope I can. Then I have to share my screen, I think, to be able to show that. I just wanted to show how this movie looks like. So this is a propionate culture, I think, but they look more or less the same. So it's been starting to degrade the acids now, and here is how it looks. So what I think is happening is that they start to form flocks and they form gas. There's sort of buoyancy up, and then it gets to the bottom again. It's sort of vacuum cleaning the whole culture. So it ends up being just one big flux. So first we saw it was like, what? Is it really alive, like an organism itself? But I think it's more like, connected to the gas formation. Yes.
From the bottom,
Yes.
and at the end up with one big.
Yeah, or often like that, but it can look a little bit different depending on the ammonia level or how many times you fed them. But yeah, it can be dispersed, so you can shake them, and but then they start doing this again. So it's really. No, I think this is. I think he did it throughout the whole. Actually, this is with yeast extract now, and I remember. So maybe 2 months or so. So it's like pictures taken, and he put them together. So.
Is it batch conditions? Yeah.
So. They will scream on Zoom in case they don't see. Yeah, I hope. So we want to look into this a little bit further. What is really the importance for them that they are connected like this in flocks? So we were thinking, well, what happens if we grow them with quite rush stirring, or if we have them on a shaking table? Because the idea with the stirring was that we will destroy the connection, while having a shaking table, we'll still have some movements in the medium, but not as rough for them. And we did some modeling of this movement, and we can see that the red part here is the shear stress. With quite a rough shear stress when you have the stirring, while the shaking is more, as we were thinking, less harsh for them in case of shear stress. And we could also see that, well, with the shaking, it was actually, these are the particles, the black ones. So it's actually collecting the particles on the bottom, sort of. So the particles or the cells in this case. So it turned out that they actually sort of concentrate in the cells with the shaking. Whereas with the stirring, you sort of stir they are sort of a little bit more gathered around the walls. And this was also seen when we just observed the communities, that with stirring we had flock formation, much lower flock formation, whereas with shaking you had these concentrational cells, they actually promoted the flock formation. So this is the results. We had on the acetate culture, we had a little bit of impact of the stirring, but it was not too bad. So, whereas with the propionate, it was super stressed by this stirring. So it was actually out of 3 cultures, it was only 1 that actually got started to grow on propionate, whereas 2 cultures, they were totally inhibited and never started. But we didn't have much of an effect of the shaking. So we thought this was quite interesting that we see a different behavior between them. I mean, they are growing with the same methanogen, but so it's the difference is that the propionate oxidizer is really dependent on having a connection with the methanogen to a higher degree, or it's stressed by other reasons. So we did some. This is actually a problem with these cultures. I mean, they are. We really want to do like our method transcriptometry in the beginning, and we tried that a lot, but they are so slow growers, so we don't get enough yield of cells. So it's a pity. And then we are working with trying to develop that, but at the moment we haven't been able to do that. So we need to feed an additional time with propionate or acetate, and then we have enough cells, have them active, and then we do their RNA extraction. So what we could, yeah.
In this case, do you use a mixed inocular or it's a co-culture of?
It's this enrichments.
Okay.
So without yeast extract. So it's the main Syntrophic bacteria and methanogen and this additional species.
Why do you think it's taking so long to get as much slower than when you use it?
Yes. Yeah, the long lag phase is so annoying. Actually, do I have no? If you remember, I think for the propionate, if I go back, maybe. I think it really connects to the first, there's many slides here. Here, I mean, what I think is that we connect the first pathway with the last one. So this is the energy demanding step. So they actually need the last step in order to start. So yeah, it's difficult for them and how to start. I think that could be one reason. So I try to feed them with, for instance, urate to sort of bring the last step up. But since it's an enrichment, there are very, very small levels of other species that I think consume the pyruvate instead. So the amino acid, aspartate, that one shortened the lag phase. So I think that one they can sort of bring here somewhere and convert it to oxalose acetate. But yeah, we need to prove that if that is the case. But I think this is also because we just actually got the manuscript with the stirring back, and it was like, yeah, but you have a very enriched community. How can you really, relate this back to the biogas digesters? And it is really a good point. It is. The problem in the biogas reactors, we have problems seeing them get enough yield now. How can you really study them in the biogas community? It's so tricky since the activity is so low. But still, these kind of things, yeah, we see the long lag phases. But this is also why you have such high accumulation of propionate, and it's so difficult to get it down because, yeah, they might not be there active, and they are at very low level. You need maybe 100 days if you are unlucky to get them to grow. So this is sort of reflecting the biogas community as well. So I mean that's I think this is one of our key things when we look into the optimization of these species is really to try to shorten the lag phase. That's something. Removing the yeast tract really increase the lag phase a lot. So there are nutrients that they need, but we don't really know exactly who and which nutrients and how much and so on. So we need to do a lot of cultivations. But with 100 days of lag phases, there are not so many cultivations you can do. So yeah.
So even after enrichment procedures, you always have these lag phase.
Yeah, I mean when we grow them in batch, we transfer them. Yeah, but then if you have them growing, if there are many cells, and then you just add the acids, then there's almost a very short lag phase. So yeah, but you can't wait for long. So if you starve them, and yeah, let's say I don't know, 20, 30 days, you feed them. If you're lucky, they will start again. And it also differs between the mesophilic and the thermophilic. So the thermophilic SPOB, they are super, super sensitive. So they are, I mean, they're very specific conditions when they grow.
And did you follow some enzymes, for example, in this pathway? Did you,
Sorry, if you follow the
different enzymes implicated in the pathway?
Yeah. So in the metatranscriptomic, we see the genes, and we can see the pathways they are taking, but we haven't done any enzymatic analysis at the moment. But that would be really nice. We would like to do that. I'm coming there later, but we have. I mean, it's also tricky because for the most of them, I mean, there are some question marks still. I mean, there are genes that are not characterized or like so. So it's definitely some question marks regarding to both species, and especially the acetate oxidizer, how it gains ATP. It's not really clear. So yeah. But yes.
Yeah. Congratulations to run batches for so long and with nothing happening for so long. My question is, in theory, if you run a CSTR, it's supposed to be washed out because they don't do, I mean, apparently they don't do anything. There are two questions related to this. Do you think that the ability they have to have a physical contact to attach to the other one way to resist to the washout, and which could be. And also the other thing is, in our experience, what we saw is in order to promote the syntrophic between organisms, sometimes you need to stress them, not because of ammonia, I mean maybe because of ammonia or something, but also like copper, zinc, or whatever, which is difficult to find. Did you see any effect of stress to the speed of Syntrophic?
Yes, but I mean, stirring apparently that stress that prolong the, but also ammonia levels that will definitely make them slower. I haven't tried trace elements. That would be interesting to. Actually, I have now. Or actually, we tried the iron and sulfur compounds, and that has also actually just prolonged them. So I don't. Most of our cultivations are done at high ammonia, so they are already stressed. So I haven't tried. I mean, actually, but when we have very very nice, but they don't compete with anyone. I mean, they are just the syntrophs, so they are the only one. There are no like extra methanogen in these cultures that can consume the acids, the acetate, for instance. So I mean, they just take their time. But I mean, if you lower the ammonia, you lower the stress, then they will be faster. So.
Yeah. What we did is we have some trace element stress lacking of trace elements. And what we saw is promoting the physical contact between the two centrops. And so I personally, I found it very romantic because when you are stressed, they kiss each other and they live together and they die together. So it's very romantic. But what we observed also later is if you remove the stress, and it's still life, they separate each other and they live their life together, and the synthol can go to another bacteria. So it's,
But then they have other substrate to consume. I guess then they will do fermentation instead. And of course, I mean, that's much easier. They will gain so much more energy by doing that. So that's definitely so. They will do that instead. So
we ran with partner in Marseille. We ran experiments in batch, classical batch, and another experiment with one bacteria within a bag where it could only exchange the liquid phase. And it was completely different. When there is no physical contact, there is no syntrophic in our case.
I want to have that bag. That would be so interesting. Yeah. But our problem is that, I mean, then you have to kill off the methanogens first, and then you and then you have the bacterial culture. I mean, that would be super interesting to do. Yeah, but I think, it's definitely so. You shouldn't provide them with too nice environments, like having a lot of different substrates. Then they're lazy, just as us. I mean, yeah. Exactly.
One question I had, when you start your presentation, you put the keywords of the discipline you're studying. And I didn't see any mathematical modeling or whatever.
You are doing that.
Yeah, but what we do, I mean, what's done, so it's mass balance, it's not metabolic modeling exactly.
Yeah, but you will do it, right? So you will start to.
I was surprised that you do CFD modeling for the shear stress or dynamics, and that you don't.
We did. It was actually a cooperation with another researcher, so we didn't do that ourselves. But actually, I had a postdoc working with metabolic modeling, and he worked with for 1 year with that. But it's I'm not stressing you now, but it's. I mean, it's complicated. I mean, he didn't have a previous experience with that, but it's also. Yeah, I mean, we have these key steps we don't really know about yet which genes are used, and there's a lot of assumptions you have to make. So we tried, but it's at this point it's it's not so easy. But I have high hopes with your experience and expertise. Hopefully we can get there.
No, but we know now also it's the dialogue between bacteria. They have kind of similar dialogue between ecosystems. And in the medical field, they could realize that the microbiota we have in our digestive tract is interacting with ecosystems related to cancer. And so microbiota exchange molecules, they found the molecules they exchange between them. So different ecosystems located in different places, not connected. And they suspect, they found evidence that they are discussing together, and the microbiota is sending signals to lower the inhibition of resistance to cancer, to some cancer. So they are
Amazing.
Yeah.
That's so interesting. So, and I am sure that we will see this kind of interaction, communication between them, definitely, when we keep on studying them. But I mean, still there's we are working with it, and we try to, but it is tricky. It's not super simple, and often you have like studied other species like E. coli and so on, and they have a special feature, and then we see the same activity in these species. We can't for sure say it's for the same reason or if it has the same function. So it's a work in progress. So communication between them is something I would really know more about. I think that's very, very interesting.
Identify if there is like specific species or genera of syntrophic propanate oxidase that can work with only specific metanogens and like not with
Yeah, this is also a very, very interesting question. And I mean, I think so. I mean, we have only found methanoculeus as a partner. So, I mean, it can't be just coincidence that it is so. I'm sure that they can probably work with other methanogens also. But, I mean, methanoculeus is also an ammonia tolerant methanogen. So I think, yeah, there must be something. I think they have specific feature that they demand from this methanogen to, and maybe, I mean, that they actually are forming some kind of connection between each other. Could be it, or yeah. So, but I would love to know more about that also. So that's something I would really like to do with this enrichment. What happens if we add an additional methanogen, for instance, from outside? So will they start to cooperate, or are they just going to compete against each other? And yeah, there's a lot of different stuff to look into. So yes.
Other questions. Looking at Syntrophic, what we realize is if you look at if you run triplicates with one species, maybe you can have different, or you can have difference in the products that are formed. Whereas when you have Syntrophic, the replicability of the results in terms of product is much higher. So you really you lower the dispersion of your results. You have more stable results at the end. Did you notice this also?
If you regarding the products, yes. I mean, they don't have many options. So of course we can see that they form both formate and hydrogen. But I mean, they can't do much other things because we only add. I mean, yeah, this is what they can do, sort of. So I haven't seen. I mean, I don't know because there might be low levels of some products, but it's quite difficult to measure them. But I have a PhD student now working with labeled carbon acids. So we are hoping, I mean, both to see about what is this one doing, and also if we can see some low levels of other products. I mean, that's also something we would really like to do.
The thing is, if you don't have Syntrophic, where the bacteria can take several routes, metabolic routes. But if you have Syntrophic, these routes are still possible, but it's much more restricted, which is good in terms of industrial application, because what you want is always to get the same results.
Yeah.
So that's one way to also.
Yeah, but it's connected to the low yields of energy. I mean, they don't have other options. They found like one possibility to go that direction. But that also makes them quite sensitive, and also I mean, it's not strange when biogas reactors you have like fluctuations. They are strictly, I mean, at least when they do this, they're quite strict in what they can do, and I don't think they can really compete with other fermenters. I mean, for the syntrophic acetate oxidizer, grows on betaine, for instance. But I think there are more efficient bacteria that do that in the biogas digesters. So, but they managed to, at least the syntrophic acetate oxidizers, I think they are competing better than the propionate oxidizers because it comes up quite quickly. If you get acetate up, they are quite quick in starting to degrade acetates. Whereas I think, at least for the propionate oxidizers, seems to be more restricted. But I don't know because I don't have them in pure culture. But I would love to look into that. But yeah. But it's definitely true, and maybe I should stop thinking about this as a negative thing. Actually, it can be quite positive in terms of industrial applications.
You said that management of the partners of Syntrophic, you have also a cohort of species that are present at lower donors.
Mm-hmm.
So where is the same species?
Yeah, I mean it's so when if we remove the yeast extract, these are the ones, acetylmicrobium and alkali. Always. And it's so interesting. The acetylmicrobium is also in the thermophilic. So this is alkalophilus is only, both of them are in the mesophilic, but they have the same metabolic feature. But in the thermophilic we also have acetylmicrobium. So it can't be. It must mean something.
And you think they can provide something that the yeast extract?
Yeah, I don't know. Because when we saw them, when we had the yeast extract, then we thought, well, it's just growing on yeast extract and maybe it's cooperating, but it's rather that they compete with them. But now when we see that they're actually active without having anything else to eat, if it uses formate, then it's competing with methanogen. If it uses acetates, then it's competing with syntrophic acetate oxidizer. But if it's actually just providing some amino acids for them, then there's some benefiting for them. I don't know. We'll see if it's good or bad for them. But we'll see. Yes. So I will just. We have some, even though we have same products, we have some variations in triplicates. They can vary a little bit in terms of weight, but it's the same products. So this is the activity when we have this. We have the first square is the wave or the wave table, and the second square is the stirred. And if they are red, they're down-regulated, and if they're green, they're up-regulated. So what we thought was quite interesting was in the stirred community, we have this oxalate formate antiporter that seems to be down-regulated. So maybe this is some key function for them when they have several methanogens close to each other or close to the bacteria. Another interesting thing was they downregulated iron activity connected to iron, both the methanogen and the syntrophic propionate oxidizer. And the acetate oxidizer, it shut down the switching motility. So this one starts to move less, maybe, and also the acetylmicrobium, both the villi and flagella sort of down-regulated that when they have movements in the medium. Another interesting thing was that we saw a shift in metabolic activity of the methanogen. It starts to circulate the intermediates, so to say, in the pathway, so it saves energy by doing that. So if it's stressed, then it has some capacity to save energy and try to survive the harsh conditions. And this is small chambers. that my PhD student has 3D printed. And we managed to get them anaerobic for a while. So, and we keep it under the microscope, and he's now trying to add ammonia to the chambers and observe if they are actually moving together. And that's the recent times he has done this, he's seen that they are actually trying to reach each other if we have ammonia in the medium. We don't know if it's like actual biological activity or if it's some chemical cause for this, but it's nice that we can actually see their actions connected to that relate to their activity. Another thing, I mean, I was saying, well, maybe flocculation is also a way for them to tolerate the ammonia better because they form flocks and maybe they can sort of protect each other by having these flocks. So we wanted to look into this. Is this important for them, and how do they tolerate the high ammonia? Since there are specific species that are ammonia tolerant. So we have done a lot of batch tests where we spike with ammonium chloride or sodium chloride. And we have done metatranscriptomic analysis after 1 hour of spiking and 5 days of spiking, because we want to see the acute stress strategies and the more sustained activities. And what we saw that I think is quite interesting is that the propionate oxidizer, it's upregulate the flagellum complex in ammonia cultures, whereas if you add salt, it's the opposite. So it starts to move if it fills the ammonia. It also starts to produce, synthesize or transport some osmolytes, and also upregulate ion channels. So these are quite known activities for ammonia tolerance. But what we also see is that it shifts the cell structure a lot. So this is the control, the green one. And this is the 1 hour after spiking with either salt or ammonia. And this is the late response of ammonia, whereas this is the late response of sodium chloride. So it really differs what it does as an acute stress tolerance mechanism. And it's also very interesting that it goes back to the control conditions, like when you have no ammonia or very low ammonia, high ammonia condition. But what you can see is that its cell structure seems to be some very important thing for it. So it doesn't change the metabolism a lot. So it's more morphology. And we are working with this data now, but I thought this was quite interesting. And then we compared with this additional acetaminophilin. And what I think is quite interesting is that one, it has a significant changes in metabolism. So, and actually, if you look at the glycine cleavage pathway, it sends around the ammonia back. It's not using ammonia, but it takes it in and takes it out. So it connects a little bit. So, and it seems to be very highly competent and very flexible. So I think that is the key here. That, I mean, many studies have sort of highlighted this as the novel syntrophic acetate oxidizing bacteria, but I'm not convinced, so to say, that it's acetate that it degrades. It seems to be very flexible, and yeah, we'll see. And the syntrophic acetate oxidized, so it's faster in adapting or didn't care much. So it seems to be easier to work with, at least. So I was saying this is just to show, I mean, they are tolerant to ammonia, but they're definitely not affected. So this is the higher level of ammonia, and we have longer lag phases and slower growth with higher levels of ammonia. So they are definitely stressed by the ammonia. So, okay, we know that, but how do we speed up the rate? So we have done some studies looking into supporting material. It's not huge impact that we can see. Maybe some positive effect of iron oxide and zeolites, but it's not. It shortens the lag phase a little bit, but it doesn't seem to be connected to conductivity. So indicating that it's probably not direct electron transfer that they are dependent on. And we see when we vary the temperature and ammonia level that we get different effects, and zeolite can also remove some ammonia levels, but it doesn't seem to be the case for these zeolites. But if you want to add zeolites for these species, it seems to be better if they're more stressed. So they benefit from the zeolite by if they're more stressed. And this I was telling just to show you. The most of the studies we have done on mesophilic cultures, because the thermophilic cultures have been so tricky. I have, like last year, finally, I've been running this reactor for 5 years, I think, because they don't grow in batch. And I have started to realize the conditions they need. So now we can start to do some experiments with thermophilic propionate oxidizers. And I think it's quite interesting because when we added iron sulfate, we see a really high activity of them. And there might be that they are sulfate reducers, but when we have the sodium sulfite, it's not the same impact. So there is something with the iron and sulfate to sort of maybe change the reduction potential or something in the community. So we are looking into this also at the moment. But you think you find a strategy for the mesophilic community is totally different for thermophilic. Even though they are so closely related, the function and their needs are completely different.
Do you think it's because of the difference in genes or expressions or whatever, or difference in abiotic effect of temperature or something?
Yeah. No, I mean the pathways they use is almost identical. So it's definitely something else. So there's abiotic effects. They're really sensitive to pH. So and also, I mean, they have other demands in terms of nutrients. Trace elements needs to be higher. So, but it's so difficult to know what it is. So you have to try one thing and one thing and one thing. So it's really time consuming to find it out. But I have a secret recipe.
You see, if you run your experiment in mesophilic conditions, so you have one mesophilic, one thermophilic, different species, and if you switch one, let's say mesophilic to thermophilic or the other, which would affect the abiotic effect, can they switch? Or
the mesophilic is super sensitive to temperature. Actually, it can't grow above 42 degrees. So that's also one thing when you operate your reactor, that you should not have fluctuations in temperature, because it seems to you can't handle it. At least not if you have the high ammonia. Maybe if you have lower ammonia, maybe. But it was not possible. I haven't tried the thermophilic and mesophilic temperature. We are doing studies now at 45 of them. So maybe. I think maybe the thermophilic will be able to grow at the mesophilic temperature.
it might indicate how it plays,
Mm.
if it has a different effect.
Mm-hmm. Mm. Yeah, that's a good point. Yeah, I will try that, definitely. This is just to show that when they degrade propionate, they have like, at least the first time they do this, is to have like a intermediate lag phase during the propionate degradation. And first we thought this is the acetate, so well, it's obviously the acetate itself that comes up at high concentrations, and before the syntrophic acetate oxidizing material can start to degrade, and that will sort of shift but it's actually not the acetate itself. It's because we tried, I don't know if I tried to add acetate at different levels, it didn't have much impact on the propionate degradation. So it rather seems to be like the connection with the methanogen somehow. So when the methanogen isn't working, or if it's working with the propionate oxidizer first, it needs some time, it seems, to start working with acetate oxidizer or something. I don't know, some redox potential or, yeah. So this is something I think is quite interesting. But when we spike with propionate the second time, we don't have this intermediate lag phase. So there is some adaptation of the methanogen that is needed for them to be able to cooperate with both bacteria in the same time.
That's an answer of the question I had. If you do 2 batches in a row, which again I know 100 days, it's long.
Mm.
Do you get the same results during the second batch? So the lag phase, for example, is long as the first batch, or it can be lower?
So I mean, if you re-inoculate in a new batch, it's a little bit depending on the inoculum and how long it's been starved. But I would say it's more or less the same often if you redo. I mean, yeah, this is what we see more or less often. But this is what we see when we have the yeast extract in the community. So when we have removed the yeast extract, it's actually not this intermediate lag phase. So I think it's much slower, and the methanogen has some more time to adapt to both bacteria before it. So it's a little bit different if you remove some nutrients. So yeah. We'll see where we'll end up. So just to show, we have ongoing some Syntrophic butyrate oxidizing bacteria thermophilic cultures. It's popped up in the propionate oxidizing community, so we managed to get it growing. So this is the first ammonia tolerant butyrate oxidizing bacteria. It's going to be nice to get this paper out. We are setting up a steady state system for SAC. So it's some initial problems. So we have medium without yeast extract. And before we use these pumps, other pumps, but they sort of pressed in oxygen in the tubes, even though the tubes should be anaerobic. So now we have some syringe pumps instead. Still getting the medium a little bit pink. But so now we are thinking of building some sort of anaerobic box here somewhere. I don't know how we manage. But it's working. So we will cultivate the thermophilic acetate oxidizers, since they are the easiest ones. And hopefully we'll get some steady state conditions for your calculations. So it's started. You just have a few months ahead of calculations. And I told you about the C-carbon, 13 carbon substrates we are doing now. And yeah, modeling and in vivo cultivations to sort of test these models. And I was also saying that in future, I would like to increase the complexity of this community. I think that would be super interesting too, because we are always struggling with this question: how do we sort of use the knowledge we have how can we use it in the biogas digester? And I mean, my feeling is that, well, if we can start increasing the complexity in a controlled manner, we can start to really see what they're actually doing, and hopefully then we will reach the biogas process.
You mentioned thermodynamics before, and and work much more simplified models politically, say much about. But we usually take thermodynamics into account, and on the graph that you showed before, where more acetate actually kind of slows down even proper degradation. For us, simply put, it would be thermodynamic.
We did calculations, and the acetate needs to be very high to actually.
It depends on the having concentration, etc.
Yeah. But yeah, so according to our calculations, I mean, the acetate needs to be much higher than we saw, thermodynamically. But we haven't had the possibility to measure formate. We could measure hydrogen, but not being able to measure formate, I think, is a lack. So now my PhD student, she's using NMR to measure formate, and it works very well. So I'm very glad about that. So that could be used in our calculations. I think that's going to be a good thing to include.
Some idea. Do you know potential of the cytochrons that you have? Nice syndromes that you have? Actually, it could be that the one that knows niche layer is somewhere. Electron transfer, maybe still some energy, small thermodynamic window if the other parameters cannot get. Something like that.
Yeah, I'm not sure, but I can try to look into that. I don't really remember about the cytochromes really, so I will go back to the data and I'll look for that. And it wasn't really something we could say for sure if I remember it correctly, but yeah, I mean, definitely there could be something there. Yeah, definitely. No, I agree. Yeah, and I just want to say thanks to your group and my PhD students and co-authors that has done a lot of work with this. Yeah, so do you have any more questions? Yeah.
I have one very general. So the slide just before this one, which was one of your first slides. Yeah, so you have an arrow going to your experiment to the digester back.
Yeah, I would say we are still doing some identification with the
yeah. If you want to favor this, what? And again, I love what you're doing. Would you favor conditions during the operating conditions of your digester in order to favor syntrophy between bacteria, like promoting the growth of blah blah blah, or promoting conditions that would favor more plastic or plasticity in the metabolic, like sarcina, with go whatever.
Yeah.
To be more resistant to stress.
I mean, that could be an option, definitely, to favor some more flexible communities. But if you want to work at high ammonia, yes, you can have some methanosarcina, but still, if you want to favor them, you need to know what actually inhibit syntrophs. Because otherwise, it's always competing. And so, I mean, if the syntrophs have gotten established, they can be quite fast. So they can actually compete with methanosarcina. But as you say, maybe it's, well, not so flexible in terms of stress conditions, or I don't know, actually. But maybe these are actually where the syntrophs are actually really, if they just get started and you have, I would say, looking at what I know about the propionate oxidizers, you would feed a little bit of propionate all the time. So you keep them active, never starve them. And then you will have the super good propionate oxidation community. But there are no operators that would like to do that, I think, or maybe.
My feeling is, if you want to have a good digester, is not to keep everything stable. You have to disturb a bit.
Yeah, definitely.
You have to spread them and favor in some occasions them, favor them, and because in the sense of they remain in ecosystems. It's not for nothing. They are on hold.
Yeah.
They can stay there. It's because they can be useful at some point. How to wake them up.
Definitely. And it's also, I mean, now I've cut the chain, but I think I'm definitely quite sure that there is interaction with bacteria doing a higher activity. So, for instance, I mean, the propionate producing communities, are they at high ammonia? Can they sort of communicate and interact somehow? Are they sort of part of a flock community with syntrophs? I don't know, maybe. We'll see. I mean, these are the things that would be really interesting to look into further. But yeah, it's tricky. You should stress them and high diversity or feed them. Or, I mean, you might have periods where you don't have any propionate production at all. So then you don't have the community that you want when it's getting other direction when they form propionates. So, yeah, after 100 days or so. Yeah.
And a very general question about Sweden. I remember 20 years ago, I visited a farm that was modified to investigate the performance of different technology for farm residues in Sweden to produce biogas.
Mm-hmm.
It was in Lund, I remember well.
Mm-hmm.
So it was very nice because you have like 10 different technologies focusing on the same waste and being able to pilot scale compare them.
Mm-hmm.
Is it still active or?
I don't think so. I don't think they have. I think no. I think I know about this, but not what I know of. But we do. We start to do a lot of. We start to work more with animal manure. It's. I was telling some of you that we have a huge increase of agricultural biogas plants this year. So it's very, very nice, and the interest for using animal manure is increasing a lot. So that's a very interesting topic for us. So yeah, I heard some of you are also working with that, so it's nice. Okay, thanks a lot for listening and for listening.